go into detail to know more about DNA sequencing. It gives us an information about how can we improve on medicine in general. Generally, like in bioinformatics, we can predict if I collect your sample, any any sample of your body, your ear, your sperms, your ovary, your blood, your urine, your stool. I can be able to predict after doing bioinformatics on your body on those samples. I can be able to predict what are the likely sickness you can be able to suffer in the next 10 years, next 20 years, concerning your age, or which one have you suffered before, or which one have been suffering you inherited from your family. We can determine even how long you can live. In bioinformatics, from those information we can access from those samples. So it is just so it can be used as a predictive medicine to determine what and what can happen to an individual. Going through different sequences, going through the biological information about an individual. And I want to talk about bit is with the creation of database like GenBank, our DNA sequencing became faster and cheaper. Bioinformatics evolved to handle the flood of data. It's now cornerstone of modern biology. Hello, can you hear me? Hello?
Yes, we can hear you.
Yes, sir.
All right. So, this thing started in 1980. How many years now? 1980. That is about almost 50 something years, right? 55. 2025, 75. 1980, 45 years. So imagine the bioinformatics I said 45 years ago, and it just came into Nigeria. People get to know about 10 years ago. So most of the things we are doing now, they have been in a system for a long time in Europe or in America. So this bioinformatics have been in a system since 1980. That's the first time somebody, delved into what is called bioinformatics. And it's a cornerstone of modern biology, which we call molecular biology, modern biology or molecular biology. And what is bioinformatics? It's about using computational tools to understand and interpret biological data. Using a computational tools to understand and interpret biological data. I can remember when I was doing my PhD, I work on sperm cells, and I did about bioinformatics of those sperm cells. And we discovered that in Nigeria, there's a lot of men that they have a series of what they call single nucleotide polymorphisms. Single nucleotide polymorphism in each sperm that can never be found in any area, in any part of the world. How do you go about this bioinformatics? What happened is that there's a lot of DNA analysis that have been done, a lot of DNA sequences that have been done. And this different type of this DNA, they discover a lot of single polymorphisms there and stored in a gene bank. What do you mean by this gene bank? This gene bank is just a database that when you do this analysis, those information, is being stored in that database. It contains about millions or trillions of data informations about different kind of individual. They work on it, on their sperms, on their here on the microorganisms. Information there is stored in that gene bank. Just like a blood bank, just like a sperm bank that be kept there. It's a database. So what you do in this bioinformatics, that when you get your result, after doing for example, do your sperm cell gene sequencing, what you need to do is to copy those results and take it to that database and check and align it and cross-check. Maybe there's alignment, maybe there's different in the gene, what's it called? The DNA sequences. So look at what you got from your result and compare, take it to a database. That database look like the range, the reference ranges. Okay, so that's what is meant by just compare it, and then from there you can see different information about an individual, what most likely could happen to someone in the future, how it can be used as a predictive medicine to predict what is going to happen to an individual. I'm just giving us general overview of what is what to understand by informatics. So it's using computational tools to understand and to interpret biological data. Biological data could be generated like we are working on your various benches, your various institutions. You generate data on a daily basis. Those data can be used to make money. Those data can be used to predict what is going to happen. How government could be even run your systems to make effective. It could be run such a way that to know the type of different sicknesses happening in every geopolitical zone of the country. But what do we do with this data? We don't generate the data, we left the data there, the data dies there, it doesn't have any effect. That is why the medics, they pick up this data, they write paper on this data, and they get promoted to become a professor, they make innovations and to get a lot of information. But we, the medical lab center, that generate this data. We left the data after doing your analysis, you keep your result there, maybe in the paper form, in the form, and it dies there, nobody uses it. These are the important information that can be used, to predict, to treat patients and to use to do so many things. Information science, this bioinformatics it's about information science. It has been applied to biology to produce a field called bioinformatics. Is to get more we call it information science. That's of course they call it information science. To informate, a lot of information are there stored in the bank, in the bank there. And how can people access this information? Is those who that study information science that are able to, to study to get those information, and they apply it in biology to produce field called bioinformatics. So biology plus information, that's what they call bioinformation. Bioinformatics. Information science plus biology called is bioinformatics. It is concerned with the state-of-art computational tools available to solve biological research problems. This bioinformatics is used to solve what? Biological research problem. There's a lot of problem we have. Like look at Nigeria, how Nigeria is being run. The government, they don't pay attention to research. The research alone can make a government to function effectively. They can predict the economy of the Nigeria using the majority of data gotten from several researches. We have in the university people that have done PhD, they've done master, they've done a lot of researches. Although most of them are copycat, they just copy change the name and the title and they submit. But there's some people that do they get a lot of grants to do these researches to be able to predict why is it that Europe and America is better than Nigeria. It's because they pay attention to research. So many research. Those research is being used to predict what is going to happen. Like for example, now in Nigeria, you can remember when we do the population of Nigeria went last in 2010. Even 2010, long time ago. And could you imagine the number of people that come into being? A lot of people they delivered, and now as I speak, Nigeria doesn't have the accurate data of human beings staying in Nigeria. So how do you plan? How do you make a plan if you don't have such information? So in bioinformatics, it enables you to access information, to be able to predict and know what exactly is going to happen in the future. The term bioinformatics was coined by Pauline Hogweg and Ben Esper to describe the study of informatic processes in biotic systems. This bioinformatics was coined out. This Pauline Hogweg and Ben Esper. For example, those classes you are asking for area of consultation, I may ask an objective. Name person, I list about 3, 4, 5 names. I may pick one of those names, and who are these people that are calling out the word in bioinformatics? It's an objective question. It can come any objectives, okay? And so you should know, and you remember that Pauline Hogweg and Esper was that those people that are calling out what is called bioinformatics, the study of informatic process in biotic systems. It is found early use when the first biological sequence data began to be shared. A development of bioinformatics as field is the result of advance in both molecular biology and computer science over the past 30 to 40 years. As an interdisciplinary field of science, bioinformatics combines biology, it combines computer science, we have information engineering, we have mathematics, we have statistics to analyze and to interpret this biological data. I will quickly show you a picture. Now, where's the picture? All right. Can you see this picture I'm sharing? Can you see the picture?
Yes, sir.
All right. Look at this picture now. These are the total combination, what made up of bioinformatics. You can see computer science at the center, you can see chemistry, you can see biochemistry, you can see biology, you can see statistics, you can see mathematics, you can see engineering. Those are those professions that comes, around and form what is called bioinformatics. Those are the things that form around those any of these science courses can use bioinformatics to analyze data. You can use this data for a predictive medicine. Okay, so the key area of bioinformatics include biological bases, the sequence alignment. Please know this sequence alignment. I think maybe by next lecture, I will pick some sequence alignment and teach us how you can read your result, the result you get, and align it with a sequence base. Okay, I'm going to deal specifically about sequence alignment. And then there's what we call gene and promoter predictions. Then molecular phylogenetics. Most of us that want to, maybe when you want to do your next year, want to do projects, you may like to work on the herbs, different herbs. What are the effects of these herbs? The ugu leaf you eat, the mushroom you eat, what are the effects? All those dongoyaro leaves, what are the effects? You can do phylogenetics on this of molecular of those dongoyaro leaves, I will just show us the little picture and we'll see how we can go about it, can study this phylogenetics. Also, we talk about structural bioinformatics. We talk about genomics, and we talk about proteomics. Proteomics means that is study of proteins, bioinformatics of protein. That's what we mean by proteomics. And when you hear about genomics is the study of information of gene of your body systems. That's what we call genomics. And proteomics, that is for proteins. And we have thousands and millions of proteins in the body systems. Those proteins can be, sequence. Like for example, when I did my PhD work, I worked on sperm proteins, and I analyzed the gene, the single nucleotide polymorphisms in those genes, and to be able to determine what time of those would have oligospermia, those would have azoospermia, those that have normospermia, and what other protein in the sperm cells. And you can analyze and predict what could happen to this person, what causes this person not to be able to, to be fertile, to impregnate a woman, for staying with a woman for 12 months with regular intercourse. All those things could be known and could be studied through molecular biology. I hope I'm communicating.
Yes, sir.
I think somebody drop a chat. Class rep, please do through the chat and let me know what it's saying. Now, what is bioinformatics? Bioinformatics is the science of collecting, analyzing, and interpreting biological data. Bioinformatics is a science of collecting, analyzing, and interpreting biological data. In exam, I may ask.
Sir, the information is just a system information, sir.
All right. I may ask in your exams, in theory questions, I may make a statement that bioinformatics is the science of collecting, analyzing, and interpreting biological data. Discuss. So when I ask such a question, one, you must give me must explain what is bioinformatics. You tell me a story, where does it start, brief history, history of the bioinformatics. You tell me what is bioinformatics, what is it being used for. You can pick an example and analyze and use for your discussions. Now you get your 10 marks. Now I say bioinformatics is a science of collecting data. As you are working in your various benches, you are working in your various facilities, you are generating data on a daily basis. The question, what do you do with this data? You left the data in fact, some of the data about 10, 20 years ago could not be even found again. It's not in the library, you can't see it anywhere. It's not being stored, no a copy of those results. How do you analyzing this data? Analyzing using statistical analysis to analyze this data. And how do you interpret this data? What are you doing with this data? This biological data you are generating. This can be using algorithms, databases, software tools. It helps scientists make sense of complex biological systems by turning raw data into meaningful insights. The data you generated can be turned to a meaningful insight. It can be used for predicting, to be able to treat, to discover new medicine to treat a patient. It is an interdisciplinary field that develops method and software tools for understanding biological data. It is interdisciplinary, as explained. It comprises of computer science, statistics, molecular, medical laboratory science. A lot of, it's an interdisciplinary field that develops method and software tools for understanding biological data. We want to look at key areas of bioinformatics. We have biological databases, repositories for DNAs, RNA, and protein sequences. Like I mentioned, GenBank. We have UniProt2. There's sequence alignment. Sequence alignment means comparing DNA or protein sequence to find similarity and differences. Like, for example, somebody is having azoospermia, or somebody is having nomospermia. Nomospermia is somebody that have normal sperm cells. Azoospermia is somebody knows sperm cell at all in those, in the sperms, there's none. So we want the DNA of those azoospermia. We want to find out what are the similarities, what are the different, what make those one in normal spermia that's not available, the type of single nucleotide polymorphism that's not present in those one that is in normal, sperms. You can compare, you can differentiate between those things using comparing the DNA sequences of those sperm cells. Another area is a gene prediction. Gene prediction that identify genes and their function in the genomes. We have structural bioinformatics, modeling the 3D structure of proteins and nucleic acids. We have phylogenetics, like talked about studying evolutionary relationships using genetic data. Phylogenetic, just like a tree. You draw a tree. Maybe, for example, you use leaves. You analyze. You use molecular data. There's a lot of components. There's a lot of phylogenetics in those leaves, and then it's like a tree, different one. How do you compare them with normal? From studying evolutionary relationship with those gene data from the one present in the gene data to determine what is the efficacy of those leaves. What do they do? Is it the number of quinine? A lot of what are the components of those drugs that's in those leaves? Then also you can talk about genomics and proteomics that I've told you. That is gene, protein. You study the gene and the protein in bioinformatics. That's what the genomics and proteomics. Analyze the entire genomes are protein sets. And then we want to look what are the applications. This information is very important. Why do we need bioinformatics? Where can we apply bioinformatics? Which field, which area do we use bioinformatics? Please pay attention to this information. Bioinformatics has not only become essential for basic genomics or molecular biological research, but it's having a major impact on many areas of biotechnology and biomedical sciences. The main use of bioinformatics include: number one, bioinformatics play a vital role in area of structural genomics, functional genomics, and nutritional genomics. What that one means is that even the nutrition, the type of food you eat, it can be determined. You can know what those food you eat, what does it take you to, what are the pros and cons of those food. That's what I mean by nutritional genomics. And then we talk about structural genomics. What do they do? The structure of those genes in your body systems. What do they do to your body? Is it affecting your body positively or negatively? Those things are that's where the bioinformatics can be applied. It plays a really vital role in structural genomics, functional, traditional genomics. Number two, it covers emerging scientific research and exploration of proteomics from the overall level of intracellular protein composition. When you look at protein profile, like I told you, we have so many thousands of proteins in the body system, so many. You look at protein structure, look at protein-to-protein interactions, look at unique activity pattern. For example, post-translation modifications, So this one is an emerging science that can enable you to study the interaction of those protein profile in the body systems. Then we have bioinformatics is used for transcriptomic analysis, where messenger RNA expression level can be determined. The messenger RNA, it can be determined. Maybe it's working well, or it's not working well, it's deficient, it can be determined. Then bioinformatics is used to identify and structurally modify a natural product to decide a compound with the desired properties and to assess its therapeutic effect or theoretically. For example, I mentioned dongoyaro leaf as an example, or many other vegetables we eat. We can the property of those things, of those leaves, can be studied in bioinformatics, and then we know the therapeutic, is it effective therapeutically? Can it be used against taking normal drugs? Like for example, ugu leaf, we all know it contains a lot of ions. What are the quantity of this iron present? What are the number of ugu leaves you need to take for you to know how it can help you to build your blood level in the body systems? All those can be studied using bioinformatics. Bioinformatics too are very effective in the predictions. Like I've said that earlier, it can be used as the predictive medicine. It can be used to analyze different type of diseases and sicknesses. It can be used for interpretations, and it can be used interpretation of clinical and preclinical findings. There's some findings you get in your various laboratory. Bioinformatics can be used to interpret those things when it's subjected further by studying bioinformatics of those parameters or data. It can be used to interpret a clinical and preclinical findings in the individuals. We want to look at other applications where bioinformatics is being used, other field where bioinformatics is being used. Number one, I hope I'm not too fast. Are you following class? I want a response.
Yes, sir. You're not too fast, sir.
All right.
Yes, sir. We are with you.
All right.
You are okay.
Now, we want to look at other field. What are the other field where this bioinformatics is being used? There are several numbers of them. There are many. I just take my time to go into details to give you a broad knowledge of where this bioinformatics is being used. Number one, in molecular medicine. In molecular medicine, human genome will have profound effect on the field of biomedical research and clinical medicine. Human genome. What do you mean by human genome? Is the gene in your body systems. Every individual have a gene. This gene is inherited from your parents, either from the mother or from the father. So with this gene, the human genome, we can predict how long you can live as an individual. For example, in Nigeria, the life expectancy of a man in Nigeria is about, I think, 47 years. What means is that a man, once you get to the age of 47 years, you've lived a normal life. If you die after that 47 years, you've lived as a man in Nigeria context. That's what they call life expectancy. And in your gene, individual from the grandparent, what inherited, we can predict what has happened. It's not by accident, normal, maybe sickness that could happen in that family. What are the sicknesses they be experiencing? What are the likely sickness they could have that could happen to them, in such in such a family? That's what I mean by the Number two, the completion of human genome and the use of informatic tools mean that we can search for the gene directly associated with the different diseases. I begin to understand the molecular basis of this disease more clearly. I was amazed when those sperm cells I study in my researches, when I go into molecular bioinformatic information database, I discovered that a human being that have maybe azoospermia or you're having nomospermia, we can predict the different kind of diseases that could happen to you as an individual from your chromosomes. We can predict what will happen to you, how long your sperm, what causes those infertility, in bioinformatics. So what it does is to study, and because these tools can be used to search for the gene directly associated with different diseases, in our body systems. Number three, this new knowledge of molecular mechanism of disease will enable better treatment, cures, and even preventive tests to be developed. In bioinformatics, we can predict, go into the gene bank to predict what are the better treatment. That's what they call telemedicine. I treated during our, just the management board conference. They want to call telemedicine. Telemedicine, as you are in Ibadan, your very location, I can consult with you based on the result you gotten. Assuming I'm your physician, somebody can consult with you from America, from UK, from every part of the world using telemedicine. And telemedicine is part of what we are using now. I'm giving lecture here from Ikeji to various destination people, students in various centers. So this molecular medicine or bioinformatics, it can help us to be able to predict better treatment. What are the better treatment you can give offer to you? The treatment you cannot get in Ibadan, the treatment you cannot get in Igbo, the treatment you cannot get in Lalate, the treatment you cannot get in Erua. I can assess those treatment worldwide to treat you, to give you a better suggestion how your sickness could be handled, or how you could be cured, or how preventive tests or preventive medicine can be given to you, or education. Please mute all the mic, please. All right, so it could be used as a better treatment or cure by bioinformatics and to prevent tests. You can do preventive tests to be developed. But then we have what we call personalized medicine. In clinical medicine, we become more personalized with the development of the field of pharmacogenomics. What we mean by pharmacogenomics is this different type of drugs we are taking. We can do, the gene, we can study those drugs and know if those drugs have, it doesn't have much side effects in our body systems. Like in toxicology, for example, taking paracetamol. In that paracetamol you take, maybe you have headache, you have pains, you have, arthritis or whatever, so, and you take paracetamol. This paracetamol you take, there's some percentage of those paracetamol that's not useful for your body systems. There's some toxic derived from those paracetamol. I'm using paracetamol as acetaminophen as an example. So we can study individual. Our body system is quite different. The way Mr. Fasasi react to paracetamol is not the way I react to paracetamol, depending on physiology and anatomy of my body systems. So this pharmacogenomics of those drugs can be used for clinical, to study individual, what are the best drug that is good for an individual. Bioinformatics can help us to determine what are the specific drug that is good for an individual in personalized medicine. This is a study of how an individual genetic inheritance affect the body response to drugs. Individually, the way Mr. Farazi respond to paracetamol, respond to aspirin, or respond to other drugs could be different, far different from my own because of our genetic, genetic makeup individually. So in personal medicine, bioinformatics can help us to study what are different drugs can be used for an individual and how do they respond to those drugs. Today, doctors have to use trials and error to find the best drug to treat a particular patient. Those with the same clinical symptoms can show a wide range of response to the same treatment. What doctor do is that when you go to doctor, a clinic, without doing laboratory investigation, without bioinformatics, get any data, they use the symptoms of others to analyze, to join. They do what is called the wrong treatment. Okay, some of those people have malaria used to have headache, they have joint pain, they could not be any, there's a lot of thousands of diseases could cause those similar sickness. That is why every individual needs to be subject to go to medical, laboratory investigation to, diagnosis to find out what actually is wrong with those patients. And they could be treated according to the result gotten thereof from those diagnoses. So, with informatics, patient can be diagnosed, and then appropriate treatment could be given to such patients. In the future, doctor will be able to analyze patient genetic profile and prescribe the best available drug therapy and dosage from this beginning. We are looking at when all the laboratory in Nigeria, they have what is called, they can be able to carry out this bioinformatics, informatics. In your table, you have, requisite, equipment to do all this diagnosis and without, struggling, without stressing, without yourself. But now as it is now, there's no light, there's no even money. Money is not even found in the hospitals to buy a latest equipment. It will amaze you that what we are practicing here in Nigeria in 2025 is long ago, over almost 60, 80 years ago in Europe, in UK. Now, for example, are you aware that there's a machine that does sample culture without no human interference? Okay, so what we are practicing now is long time, we are not current with what is happening in Europe. So in future, doctor will be able to analyze this. If you can get genetic profile of an individual, they do test they do to get a genetic profile of an individual. They can prescribe the best available drug therapy that could suit. I can treat that patient within less than 24 hours. We have bioinformatics can be used also as a preventive medicine. With the specific details of the genetic mechanisms of diseases being unraveled, the development of diagnostic tests to measure a person's susceptibility to different diseases may become a distinct reality. So, bioinformatics can be used to treat a patient, everyday patient, as they come into the hospital after being properly diagnosed. And that's what they call gene therapy. Bioinformatics can be used in gene therapy, number one, in not too distant future. With the use of bioinformatics tools, the potential for using genes themselves to treat diseases may become a reality. So it is possible for a gene to go to gene bank and use that gene, to treat a patient. To treat a patient after discovering after diagnosing them for various diseases. Gene therapy is the approach used to treat, cure, even prevent disease by changing the expression of a person's gene. Like those who have SSS, their gene could be replaced. They can produce a new gene to treat them, to clear off those sickle cell blood in their body systems. Drug development, and that way bioinformatics can be used. Drug development. At the present, all drugs on the market target only about 500 proteins. With an improved understanding of disease mechanisms and using computational tools to identify and validate new drug to target more specific medicine that act on the cause, not merely the symptoms of the disease, can be developed. What we treat in Nigeria is symptoms. You go to hospital, they ask you what happened, what is your complaint? I have headache, I have body pains, I have tooth pains, I have neck pains. They base their treatment on the symptoms. Then they are, in the future, we are looking at a computational tool, just like a computer, to identify and find new drug targets in the body systems using bioinformatics. This highly specific drug promise to have fewer side effects than many of today's medicine. So if you use bioinformatics, this computational tool, to identify, to diagnose a patient, it's more precise, it's more accurate. The accuracy and precision is very, very high, that those drugs will have little or no side effect. We are hoping we get there someday. Also, bioinformatics can be used in microbes genomes applications, microbial genomes applications. The arrival of the complete genome sequence and the potential to provide a greater insight into the microbial world, and its capacity to have broad and far-reaching implications for the environment, for the health of an individuals, the energy, and the industrial applications. For this reason, in 1994, the U.S. Department of Energy, DOE, initiated what is called Microbial Genome Project, 1994, to sequence genomes of bacteria useful in energy productions. Are you aware that some bacteria that are used to produce energy in U.S., in America? Bacteria. There's some area, bacteria bank. They harvest those bacteria in large quantity and producing energy. They use it as a source of energy. It can be used as environmental cleanup. Do you know in some country, they don't have what is called mosquito? Why? Because the gene of those mosquitoes have been affected. After affecting this, the gene, and they produce something similar to that mosquito to clear up in China. They clear up all the mosquitoes in the country. Okay, so that's the work of the microbial genome applications. Also, industrial processing and toxic waste reductions. Most of the toxic waste we generate in Nigeria, and nobody knows, it doesn't have it, nobody thinking about it. It's a money-making venture that you can use, microbial genome, this project, to remove the toxic waste, to reduce them totally in these systems. Like look at the forces oil. About replacing the motor. When you start your motor, those motors are smoke. See smoke everywhere. People are inhaling, toxic every day that affect their body systems. Or maybe those with where the MTN mass cell is. There's a lot of, toxic that have been generated, radiation that's coming out from that MTN mass. So if you have MTN mass in your body, in your environment, gradually, those things is being absorbed to your body systems, not through your nose, through your skin, through every part of your body, every day, every minute, every seconds of the day. And once we tend to generate, to discover, to get what is called cancer, but this toxic way can be reduced, by way of applying bioinformatics in microbial genome applications. By studying the genetic material of these organisms, scientists can begin to understand these microbes at a very fundamental level and isolate the gene that give them their unique ability to survive under extreme conditions. I remember now something just dropped now. Our HOD, Dr. Akila, okay, he discovered one bacteria in his PhD thesis. There's a bacteria he discovered in southwest Nigeria that has never been discovered before. Why? Because of this bioinformatics. He discovered it, and we present. An article concerning this microorganism. I couldn't remember the name of organism now. There's one. Who is that? Please mute such student. Please mute such an individual. All right. So this discover a new microorganisms in the southwest Nigeria. I couldn't remember the name now. So, and this why, due to the course of bioinformatics, new thing could be discovered. Another way or important of bioinformatics is waste cleanup. Waste cleanup. There's this bacteria called Deinococcus rhododendrons. It's known as the world's toughest bacteria. Deinococcus rhododendrons is known as the world's toughest bacteria. Please mute your mic. Please mute your mic. And it is the most radiation-resistant organism known. This bacteria is very unique. It cannot be easily killed. No radiation, no heat can destroy this bacteria. Its name is called Deinococcus rhododendrons. Scientists are interested in this organism because of its potential usefulness in cleaning up waste that contain radiation and toxic chemical. So this particular Deinococcus rhododendron can be studied genetically and see why is it that it has a resistance to radiation. The no amount of temperature could kill this bacteria. It could be studied and then find way to eliminate this particular organisms. Can be used as waste cleanup, in our society. Also, that what they call where bioinformatics can be used also is climate change studies. To talk about climate change. I'm going to tell you now why is it happening. When is it that rain? We cannot predict when rain will fall. When rain is supposed to be falling and consistently, things have changed. Why? Because of the climate changes. Increasing level of carbon dioxide emission, mainly through the expanding use of fossil fuel, that is petrol, for energy, and are thought to contribute to global climate change. Most of the things that is being emitted into the environment, this is different kind of people. When I was in Port Harcourt, there's a particular people. A lot of people, they do what is called bunkering. While they break the pipeline, they will take the crude oil and begin to cook it live. This petroleum, when you boil it at high temperature, it begin to, there's what they call fractional distillation. PMS they say quota. And as they burn this thing, you see a lot of things emitted into the air. If you park a car outside, a white car or any car outside, within 4 or 5 hours, you see a lot of things on it. Black soot, we call it soot. You can hear, went to my phone, you see, it'll be black. Why? Because a lot of emission, a lot of things that are cooking every villages everywhere across the Niger Delta. And they never knew they are causing havoc, and those things cause what is called climate change. While in rain will fall in no stop, it can fall like 7 days. A lot of things happens. Why? Because of this emission of soot, of carbon emission into the climate. Recently, the DOE, Department of Energy in USA, launched a program to decrease atmospheric carbon dioxide levels. One method of doing so is to study the genomes of microbes that use carbon dioxide as their sole carbon source. What we are saying is that there's a particular, microbes that use carbon dioxide. These microbes could be affected. If you want to reduce carbon dioxide, that is carbon dioxide you want to breathe out of the body system. It can be used, to reduce the carbon source in the atmosphere. Also, another way it can be used alternative energy source. Scientists have studied the genome of the microbes, Chlorobium tepidum, which has an unusual capacity to generate energy from light. These microbes can be used to generate, like I told you, there's a bacterium can be used to generate energy. Also, there's other genomes and microbes also. This Chlorobium tepidum can be used. It has the capacity to generate energy. We can use in the homes. We have power problem in Nigeria. Just imagine some of these microbes is being generated harvested in large quantity, in millions, and supply a community or supply a city with light. How it's possible, we get light through, apart from the water, from the the dam water, can get light from the solar. Thank God most of us are using, solar panel, now when there's no light, there's a microorganism. Wind can generate energy. There's some country they use winds to generate energy. Now, another information is that the microbes can be used to generate energy to give us power supply. All right, so it can be used as a form of alternative energy source. Also, biotechnology. The Archaeoglobus pugedus and the bacterium Thermotoga maritima are potential for practical application in industry and government-funded environment remediation. These microorganisms thrive in water temperature above the boiling point, and therefore may provide the DOE, Department of Defense, and private company with its stable enzymes. It is suitable to use in industrial process. This bacterium I mentioned, they are very, very, they can survive at high temperature, above 100 degrees centigrade. They live there, so they can be usable to use for industrial process for its stable enzymes can be used in industry. This substance is employed as a source of protein in animal nutrition. This substance, this microorganism can be used to produce protein for feeding animals. So biotechnologically produced lysine is added to feed considered as a source of protein and is an alternative to soya beans or meat or bone meals. Biotechnologically, with informatics, there's other sources for protein can be used without using soya beans, without using meat or bone meal to add it to animal food. There was another bacteria called Lactococcus lactis. It's one of the most important microorganisms involved in the dairy industry. Although researchers anticipate that understand the physiology and genetic makeup of this bacterium will prove invaluable for food manufacturers as well as pharmaceutical industry, which is exploring the capacity of lactase to serve as a vehicle for delivering drugs. We have antibiotics resistance. Scientists have been examining the genomes of Enterococcus faecalis, leading cause of bacterial infections among hospital patients. Like this is a project topic for someone. You can in your hospital, you can sample patient, you can collect samples, stools, maybe 200 samples or others are patients, collect their stool sample and analyze for this particular bacteria, or is a common cause, a cause infection in hospital patient. Is it available in your hospital? It's not available. It's a project topic, so you can look at it and see whether this Enterococcus faecalis could be found in your environment. So bioinformatically, antibiotics, you can examine all those bacteria that may be resistant to antibiotic treatment. They have discovered a virulent region made up of several antibiotic-resistant genes that may contribute to the bacterium transformation from a harmless gut bacterium to a menacing infector. This discovery of the region, known as pathogenicity island, could provide useful marker for detecting pathogenic strains and help to establish control to prevent the spread of infection inward. In forensic analysis of microbes, scientists use these genomes to help distinguish between the strain of Bacillus anthracis that was used in summer of 2001 terrorist attack in Florida with that of closely relaxed anthrax strains. All of us, I don't know whether we heard the history of, the attacks in Florida in 2001, terrorists. They picked these particular animals, to cause infection, to spray into the air, to cause a lot of to attack an individual in America in 2001. The terrorists, they use it to attack. I mean, so it could be, this microbe could be analyzed and studied and used this particular anthracis to know and to produce that similar bacteria. So if such an individual have that infection, it could be used as antidote for it. A vaccine can be produced to use against such an individual that have been infected by this bacillus and anthracis. Also, bioinformatics can be used. Also, the reality of bioweapons. Do you know that there's some viruses that are used as bioweapons? Bioweapon means please mute your mic, please. So what am I saying is that these bioweapons, there are some viruses, there are some bacteria that could be used. Please, can you mute those mic, please?
Please, user. Let the user mute his mic.
Okay. Can you hear me, please?
We can hear you, sir.
Yes, sir.
That person is interfering. Mr. Francis, please mute all the mic, please.
Francis, you recognize that person. Meet him or
see
her.
the person here.
You, sir.
Please mute your mic.
Please let them remove everybody.
All right, let me continue. Sorry for the polite interruption. The person is still talking. So there are some organisms, that can be used as a weapons. We call those organisms bioweapons. For example, we want to destroy some community, like this Boko Haram, this jihadist. You can use what we call bioweapons to finish them. One, this like polio virus. Some virus can be affected, and then used as a weapon from here and spread out their particular locations. Every human being that's staying in that environment, every animal that's in that environment, they will be infected with those viruses. We can terminate their life within a short period. So scientists have recently built a virus, for example, polio virus, using entirely artificial means. They did this using genomic data available on the internet and material from a mail order chemical supply. The research was financed by U.S. Department of Defense as part of the warfare response program to prove to the world the reality of bioweapons. What I would say is that there's some organisms that can be used as a bioweapon to fight any individuals, So, and American defense, they use this weapon, they invest so much on informatics to generate what is called biowarfare. They use this to generate bioweapons, to attack, to store them. Okay, maybe there's a war they want to fight. They go to the bioweapon banks and then begin to, get those organisms and use to infect any particular area or any country, any nation they want to attack. So the researcher also hope their work will discourage officials from ever relaxing program of immunization. This project has been met with very mixed feelings using bioweapons. Like, there's some country, they have nuclear weapons to eliminate, they want to eliminate the whole world, the entire world. Within less than 2-3 days, they can eliminate the entire world. They're using nuclear weapons. nuclear weapon, when you just excel, no, such an individual is not inhaling. Once we inhale, when they contaminate the air, they can eliminate the entire world, about 8 billion people within 24 hours. So also the bio weapons also can be used, to fight. To fight in a war with the help of bioinformatics. We have crop improvements. Comparative genetics of plant genomes are shown that the organization of their genes has remained more conserved over evolutionary time than was previously believed. Like for example, I remember when I traveled to China 2011, 13, yeah. There's a time I saw a corn, Agbado Tanje. Agbado doesn't have a season in China. They eat corn almost every month. Comes out. Why? Because it's one of the GMO food. That genetic modification food. Could you ever think that in Nigeria we can be eating corn? There's no season for corn. Most of the corn we planted, they can be genetically modified to can grow at any time. So what are we saying is that the crop, there's a lot of crop improvement. There are some crop that can grow. Have you seen that orange? You can grow an orange now. Under 1 to 2 years can be affecting your orange. Remember the type of old orange we have, you plant orange, it may take like 10, 20, 15 years before you begin to harvest orange. But now, because of it can be genetically modified in such a way that those things, their lifespan, their production age could be affected. Could you imagine how the cocoa tree can be planted, within 1 or 2 years and begin to harvest cocoa in large quantity? Have you imagined that cocoa can be planted and be stunted growth? You don't need to be low a quarter of an acre. If it's not in such a way, you can use your hand. If it grows in such a way that it produces enough seed, and this thing can be improved using biological, bioinformatics, informatics, yeah. The insect resistance, improve nutrition quality, the nature of food we eat. It can be genetically modified. Those food we eat, can be modified such a way that it contains so much vitamins, iron, and other micronutrients. Also, we can produce iron could be added to some of the food genetically to improve our hemoglobin in the body systems. It can be used in development of drought-resistant varieties, veterinary sciences, and lastly, why is it matter to use bioinformatics? One, briefly, summary of what we just discussed: medical research. You can identify disease genes in drug discovery, targeted proteins with precision. Agriculture, like I said, it can improve crop genetics, and then personalized medicine. It can be individual drug can be tailored for individual treatment based on genetic profile. And that is how we come to the end of today's class. Thank you for listening, and God bless you. Do you have any questions?
Amen. Thank you for this. Wow.
You can unmute. Please signify I want to ask questions. So, Mr. The class can unmute you, and then you ask a question. Indicate if you have, by raising up your hand, if you have question, then the class will unmute you, so for you to ask a question. Any question?
My hand is up. Oh.
Okay. Are you? You can ask a question, sir. Go ahead, sir.
Okay. Good morning, sir. Thank you for this beautiful presentation this morning. Yeah, I want to ask questions about the use of biological agents to fight some war. I want to ask, is there national and international regulation about this, or we are just free at our own, willing, that if you just feel that there are some group of people somewhere that are not doing what is good, and we can just pick some of these organisms and then go ahead to fight them? Thank you, sir.
Okay, thank you for your excellent questions. No, that is why in America, America has a lot of security and a lot of technologies. They can monitor what is happening every part of the world. They can check what is happening. They have what's it called? They have a facility to know what is even in your room. If Donald Trump decide to know who you are individually, now know your color. It can picture your room, know what you are doing at every particular time in time. That was how they managed to kill Osama bin Laden. So what am I saying is that there's regulatory agencies like in Nigeria. I don't have much information about Nigeria, but in America, they regulate what is happening. That is why they always have issue with countries that use nuclear weapons. They give them restrictions. There's some restrictions. They don't say you cannot have the tank. They don't say you cannot have nuclear weapons. There are some international regulation agencies that regulate what you have in your country. Also, these bioweapons, also, there's an international regulation agency. They regulate, they monitor, they see what to do. There's a control over all those issues. But there are some still countries that still hid their information away, except if America decides to know what is happening. They have a satellite to monitor everything happening in every part of the world. They've grown to that extent. Imagine 2000, for example, in 2000, is this a classical example? The 2017 or 16, I traveled to America. So mistakenly at the point of entry in Atlanta, Georgia, they asked me, "What have you come to America to do?" I told them mistakenly. Most of the travel I travel for conferences. I just told them mistakenly I've forgotten what took me to embassy before I got the American visa and said I came for a conference. The immigration officer looked at me, what? He just picked my international passport, called one, and that immigration officer put in a red file and took me to somewhere, a room. I was there for more than 1 hour, questioning me, check my phone, check my WhatsApp, check my email, every email I've ever had. They check and scrutinize me totally. After checking my everything, they discover nothing hidden at all. So they let me go. I was heading to New York City. So on coming back, I forgot the incident. On coming back, as I got to the airport, what they do, they just take one application, they rub my finger and my hands, my two palms, and they just he told me sorry in the next 2 minutes. What amazes me is that they check, there's the equipment check if I've ever handled ammunitions during my stay in America. I was surprised. So what am I saying is that they have a latest technology to monitor what is happening, to regulate what is happening across the world. Okay, so it could not happen. They know what is happening. They know there's a satellite. Like there was an incident that happened in Nigeria last year, something when Buhari was a president, that they kidnapped some Americans. Do you know America got to Nigeria territory? Nigeria never knew. Secret, nobody knew. They came, they rescued them, and they took them away before they announced it. So that's how powerful, how America, and those things could be regulated by America. I hope your question has been answered.
Yes, sir, definitely. But sir, sorry, sir, with these challenges of insecurity we are facing, what's holding our government back in employing some of these technologies to, solve these problems, sir?
That's political. That's a political question. Well, what is happening? One president said, if any insurgency happen in Nigeria more than 1 week, more than 24 hours, that means Nigeria government is involved. So I want to assure you what is happening in Nigeria is involved by government, and they know. Them people are making billions. They never wanted to stop. If governments decided to stop insecurity in Nigeria within 30 days, I can assure you, Bola Ahmed Tinubu can stop insurgency within 30 days. If just, I'm telling you this is honestly speaking.
Thank you so much, sir.
So that's it. Thank you.
Yeah.
Any other question?
So well done, sir. Thank you for the lecture. My question goes as you've highlighted to us so many areas in which bioinformatics have been very, very helpful to the whole mankind, especially in the area of GMO, genetically modified organism foods. But I want to ask this question, sir. I want to know why people generally have this phobia for GMO foods. Because I see even in the advanced country, some will still label their product and producer product are GMO free, GMO free. I think there's something we did with this GMO. I remember these people that promote GMO, But again, these people in America that sells all these chemicals and the rest. So I want to know what actually do you think can be that could be X risk for this GMO food that makes people to be skeptical about consuming them despite their numerous benefits as highlighted by you, sir.
Okay, thank you so much for your excellent or wonderful questions. The GMO food, it has advantages and disadvantages. One of the advantages is that it makes food available at every point in time. Two, it reduces the lifespan. I mean, maybe the time of production of those food. For example, if you plant a maize, maybe maize takes 3 months for you to harvest some maize. It reduces the time of harvesting that maize, maybe to 2 weeks or 3 weeks. And what happened? What is genetic modification? Is because of the gene of those particular crops have been modified. Okay, and those genes that modify, they have side effect, biological effect on the body systems. One of the advantage of those genetic modified food is that they have a clinical implications in the body systems, the cells, the tissue, the organs, they react to it. That is why you see in America, the rate of cancer is in millions. The cancer, because those people, most of the food, about 80% of their food, they are genetically modified foods. So the only thing you can do is to avoid, and in America, if you go to the store, those things are being labeled. If you are taking genetically modified food, it's been labeled, be written there, GMO food. But the problem of Africa is that when you pick up something to read, we find it difficult. One man said, if you want to hide anything from Africa, put it to written, write it down. They won't read. So it's clearly stated on those genetically modified food that this is a GMO food, this is not GMO food, it's stated. So they are very careful. Some people, they don't take it at all. There are some people that are vegetarian, they take only vegetables, nothing, no meat, nothing. So it's one thing is to avoid taking them after going through those content of those products. Thank you.
Thank you so much, sir. You're welcome, sir.
Any other questions? A lot of people have dropped. They are not interested. A lot of people have dropped.
Lecture is very interesting, though. It's not good to meet self.
No, sir, we are with you.
It's true.
Hello, sir.
Interesting.
Yeah, go ahead, sir. Go ahead. Any question? Your question, please.
The lecture is very interesting.
Dr. Ken Adegoke, I agree to you, sir.
Hello.
Yeah. Good morning, sir. Thank you for the lecture. Really enjoyed it.
I'm very loyal to your government, sir.
You are welcome, sir.
Yes, sir.
Yes, sir.
So, in absence of okay, let me just repeat let me recap. Let me recap what I just said about exams. And for most of us that came in late, I can assure you, that nobody will fail in this class. Nobody will fail in this class. I can assure you that.
We'll try our best.
We make it. We know too well that this is just more or less an adult education, except somebody that want to have a first class. That's you can have first class. It's a added advantage. But as much as we are concerned, everybody, we pass in this class. So please just go through those notes. We've not concluded on the mode of the questions are going to be set. We lecturer we meet, we discuss on how, and we brief you maybe 2 or 3 weeks before your exams. We update you on the model. The pattern of the questions. Like in my school, like I told you that came in earlier, that in my school, if you have 2 unit courses, 2 unit courses is objective question is 30 objective questions. Objective question is what we call MCQ, that's multiple choice questions. Why the theory of 2 unit courses they will set 5 questions for you to answer 4. And each of the questions there are 10 marks. It's be stated there. Why 3 credit unit courses are objective are 20 questions for I think 30 minutes, and then the objective, the theory is it says 6 questions for you to answer 5 questions. They are equally 10 marks. Right. So the program is projected. We know you are very busy. We know it's another class. We know you have a lot of distraction, a lot of engagement. But I can assure you that nobody will fail in this class. 2, Then what they call continual assessment. Continual assessment make up 30 marks out of the 100 marks. It's your attendance to lecture. That's why attendance is very important, And then you give text, may write text, and then assignment given to you. It contains 30 marks. An average student should be able to have A. Why? If out of that 30 marks, see, if you have something like 25, 28, and then your theory or your exams, you get 50, you've gotten A, or 40, you've gotten A. So that is very, very easy. So please, let us try and go through those lecture notes, but we can assure you everybody will pass. So no panic, no fear for anything. We make sure everybody move to the next class. And by this time next year, be preparing for our convocation, be preparing for our graduation. Most of us will be coming, to the end of our project, and then all of us are good to go to get the degree we deserved. Thank you, and God bless you. So let us be attentive and wait for further information.
Thank you, sir.
We have come to the end of this lecture. Thank you, and God bless you. Do have a wonderful weekend.
Thank you, sir. God bless.
Thank you, sir.
Thank you, sir.
Thank you, sir. God bless you, Rich.
Thank you so much.
Thank you. Able lecturer.
Power. Táwọn ní wọn ṣe rí ìwé kan.